morning, everyone. It's my pleasure to moderate today's webinar, and it's a great honor to introduce today's speaker, who is also my friend, Dr. Wayne Newton. Dr. Newton will talk to us today about a very important topic in molecular virology, which is how to assess the performance of molecular viral load assays in the clinical lab. A little bit about Dwayne. Dr. Newton is currently a scientific affairs manager in quality systems at Bio-Rad Laboratories. Before that, or before joining Bio-Rad, Dr. Newton enjoyed a 20+ year career as a clinical microbiology lab director in both public health and academic medical center labs. Most recently, Dr. Newton served as the director of the clinical microbiology lab and the associate director of the clinical pathology at Michigan Medicine, Ann Arbor, Michigan, and also held faculty appointments in the medical school and the School of Public Health at the University of Michigan. When interests and expertise are in developing and executing the strategies to optimize the impact of lab diagnostics for the improvement of patients' care. So it's our pleasure to have you, Dwayne. But just before we start the webinar, I just would like to remind everyone to please stay on mute. We will answer questions at the end of the webinar via either the chat or the question and answer buttons at the bottom of your screen. For any reason, if you lose your connection during the webinar, you should just go back to the login information and try to log in again. If you have difficulty with audio, you may try to connect by phone. An email will be sent to everybody who participated in the webinar after the webinar with the evaluation and information on obtaining the credits. If you don't need to complete, the evaluation if you don't need the base credit. Also, the webinar is being recorded, and information on how to access the recording will be sent after the webinar. Also, on behalf of the Pan-American Society of Clinical Virology Educational Program, I would like to thank Bio-Rad for their support of this webinar. So, Dwayne.
Thanks, Dr. Mustafa, for the introduction. I want to first thank Jess Warner for all of her work in helping to get this coordinated, and to PASCV for the opportunity to speak. And last but not least, I want to thank you all for taking time out of your day to join me and us today and listen to this presentation. Some of the information I think is maybe a little bit of background, but maybe being presented in a different context. So I'd like you to think about some of these concepts a little bit differently, and hopefully give you some food for thought on how to approach quality assurance in your laboratories with regards to molecular testing. And I think first and foremost, recognizing that the clinical requirements for diagnosis and management of many viral infections dictate the performance levels that our viral load assays need to achieve, regardless of whether those assays are laboratory developed or approved by a regulatory body. In the presentation I'm going to give today, I'll discuss how Unity real-time data management software from Bio-Rad can be a useful tool for monitoring our molecular infectious disease QC data, analyzing assay performance, and ensuring that we're fulfilling the clinical requirements of performance for our viral load assays. So as a disclosure, as Dr. Mustafa mentioned, I'm an employee of Bio-Rad, and I'll be talking about one of our products today, but I'll also be talking about a study we did in collaboration with colleagues at Michigan Medicine. This presentation itself was adapted with permission from a presentation originally developed by my friend and colleague, Mike Bachman at U of M. As an outline today, I'm going to provide background on how viral load assays are used. I'll describe the clinical rationale for establishing parameters for acceptable assay performance, and discuss a variety of sources of errors that could affect our assays. I'll also provide some highlights of the study that I just mentioned that we published this year in Journal of Clinical Microbiology. We did that in collaboration with folks at Michigan Medicine, where we use Bio-Rad's QC data management software, Unity Real-Time, to capture QC data, measure sigma values, and calculate quality rules in the context of the molecular assay's total allowable error. Lastly, I'll present a few important takeaway points that may be helpful to you in relating this information to the processes that you utilize in your laboratory. So first, we'll talk about viral load assays. And for you all, I know it's pretty clear that there are a variety of indications for the use of viral load assays, each of which are going to vary depending on the pathogen and the type of infection. So viral load assays can be used to make decisions on initiation of treatment for chronic infections like HIV, HCV, and HBV, can be used to detect reactivation of latent viral infections like CMV, BK, EBV, particularly in transplant patients. Viral load tests can be used as an adjunct for primary diagnosis of infection where serology is the primary tool for diagnosis, such as HIV and EBV. But in all these cases, the viral load assays are used for monitoring of treatment and looking at how the patient is progressing in the context of their therapies. And when we perform our tests, these are, as you know, are quantitative tests, but we need to determine what kind of change is happening and whether that change is clinically appropriate and significant. And one way to do this is by looking at patient values over time and looking for relative and relevant changes that occur in their viral loads. The frequency of that testing really depends on how quickly the virus is cleared under normal conditions. So as an example, in the case of CMV, that's about 3 to 8 days. So testing any sooner than that could falsely indicate persistence. One of the challenges with this approach, though, is that you have to use some math to determine how much of a change has happened and then apply that math to determine whether that's clinically significant. Another approach to take to determine that a difference has occurred is establishing a threshold. This one's a lot easier. It doesn't require any math. You just set a level. It simplifies the interpretive guidelines, and it is easy to teach and memorize for the clinicians that are using it. However, it assumes that there's no bias between the assays that are being performed for this test, the instruments that are being used, or the laboratories testing for the same analyte. And one of the challenges, as I know you're aware, is that if you establish your threshold at, for example, 10,000 copies per mil, rhetorically, is 10,001 clinically significant and 9,999 not? It's a rhetorical question, but I hope to present some information today that'll help you determine an answer to that. To illustrate how these two approaches, the relative change and the threshold approach, can differ clinically, I'm going to share with you an example of an assessment that was performed while I was at Michigan Medicine, where we were trying to determine the comparability or commutability of two different BK virus viral load assays. So the lines that you're seeing here are values from the same patient, but tested on two different assays, assay A and assay B. So the assay A was an assay that was sent, or the samples that were sent to a reference lab, but were split and tested by the assay that we were performing in-house. And you can see that there are absolute differences in the values that were obtained between the two tests. But interestingly, you can see that there are similar changes in the trends over time. So if you apply in this situation, I'll try to turn my pointer on here, you're seeing these similar trends up here. Unfortunately, we didn't have enough sample to be able to split and test here, but for other patients, we were able to see these similar relative trends where when when one was going up, the other one was going up. When one was going down, the other one was going down, but there was still an absolute difference. So if we're looking at relative change and using that to determine clinically significant, both assays were performing very similarly. But if, for example, we use 1,000 as a cutoff, you can see that the blue assay reach that threshold sooner than the orange assay. So the challenge here is what to use and what to choose to act upon. Again, this is a rhetorical question. It's just showing that in this example, there's difference, and we can't say just by looking at it, which criteria should be used. So this is just highlighting the challenges that exist when you have assays performed in different labs that are not commutable to each other. Another approach that can be taken is just looking at detected versus not detected. So it's not a quantitative difference, it's a qualitative difference. And this is really relevant for HIV and HCV because for these viral load assays, as not only therapy has improved for both of these infections, but the analytical performance of the assays that are available has really, really improved. And we can detect very low levels of these viruses. So detection versus non-detection can help distinguish successful treatment from non-compliance or resistance. But we have to remember that, especially for HIV, transient low levels of virus may occur. Those are referred to as blips, and they may not be clinically significant and need to be confirmed. So we can establish potentially two thresholds, detected or non-detected versus a relevant change or relative change over time. I'd also like to share again another example of a real case that we were presented with during my time at U of M that highlights additional challenges in assessing clinically significant changes. So this is a case of a 49-year-old man who had traumatic brain injury and acute myeloid leukemia that was admitted with some neurologic symptoms, impulsivity, and hallucinations after a bone marrow transplant. Because of the neurologic issues, CSF samples were collected and a variety of tests were performed. Some of the core lab tests showed elevated protein and a low white blood cell count. Infectious disease testing included HHV6 viral load, which showed 1,100 copies per mil in the CSF and concomitantly a 29,000 plus copies per mil viral load in the plasma. So for presumed HSV encephalitis, HHV6 encephalitis, the patient was started on foscarnet. So the clinicians chose to monitor plasma viral load over time as an indicator of the patient's respond to therapy for presumed CNS disease. And you can see that when therapy started, the viral load went down and then it went up. And so there was a question of resistance, and then it went down again. And the clinicians wondered, was the patient really responding to therapy? So there was some concern about that increase in viral load and whether or not this was resistance. But I think it's important to remind ourselves that we need to think of these assays on a log scale. Viral loads are quantitative, as we know. Real-time PCR calibrated to standards with known concentrations, and PCR as an exponential function ideally will result in a doubling in the amount of product each cycle. So detecting less than a twofold or one cycle difference in values is really pushing the limits of technology. So a twofold change mathematically is equivalent to a 0.3 log change. A threefold change is equivalent to about a half log change, and a fivefold change in arithmetic values is equivalent to approximately a 0.7 log change. So the previous slide, I presented the data as copies per mil, but when presented as log copies per mil, you see that there really isn't any change in the values. And when looking at it further, it was recognized and determined that the patient actually had chromosomal integration of HHV6, and that was what was being detected in the viral load test, and it wasn't an active replication. And the symptoms in this patient were ultimately attributed to an underlying traumatic brain injury. So again, we need to think on a log scale so that we can best understand what a clinically significant change is and in the context of the precision of the assay. So while our desire is to always be able to use viral load assays in the way that I've described, there are a variety of issues that we know can occur that cause errors and have the potential to impact the quality of the results that are being reported. A couple of different types of errors are random errors and systemic errors. So random errors could be an error in a manual step, an air bubble in a well, and those types of errors can be resolved easily by repeat testing. And systemic errors are changes that cause values to be consistently higher or lower than the true value. These types of errors require investigation and even may require recalibration or service of your instrument. And ultimately, those types of errors, the systemic errors, have the potential to impact patient results and are really the type that we're most concerned about identifying.
Dwayne, before you move on, your calendar is popping up on the lower right.
Sorry. Thank you.
Thank you so much.
It's telling me that I need to be here right now, I think. So sources of the error are extraction efficiency, thermocycler issues, or optical calibration. So performing or extraction efficiency can be have an impact where we're providing more or less DNA in the assay than is used for the calibrators. And so if we recalibrated that the assay each run, that would be an optimal way of correcting for that imperfect efficiency. Another issue that could arise is that there are changes in the thermocycler where the temperature changes or rate of change between steps is affected. And that could result in erroneous results. And there may even be problems with optical calibration that changes how the instrument detects the amplification of a template. So different manufacturers can account for different of these sources of error, in particular extraction efficiency, in different ways, but we just need to be cognizant that these errors can occur. Reagents problems can also be sources of error. So control concentrations where we have an undocumented change in lot or decay of sample can affect the actual values that are generated for the controls. While this wouldn't have an effect on individual patient results, it can result in inappropriate passing or failing of runs. We might have issues with enzymes or other reagent contents where an undocumented lot change or decay of reagents occurs. Those types of issues could have significant effects on patient values. There may even be some issues with the calibrators where there's differences between the assigned value and the true concentrations that are being detected because of some degradation, or an undocumented lot change that is not recorded. So we're expecting one value, but really we're getting another. Again, the errors with calibrators can have significant effects on patient values. So some of the impacts that I think we're all aware of these erroneous results are delayed or current use of unneeded initiation of therapy because we have false results, either false positive or false negative. Delayed detection or false detection of reactivation of latent viruses, delayed primary diagnosis of infection, or false reassurance or false concern about, about response to therapy. These really all have a huge impact on patient results, and not detecting a change that happened or inappropriately detecting a change can have significant impacts on how that patient is going to be managed. Ultimately, there is a cost. There's emotional costs, there's operational costs, and there's financial costs to these types of errors. So delaying results for all patients being controlled if you have inappropriate rejection of runs, rejecting a run because of that erroneous result has an impact on your efficiency and your operational efficiency. You have to go back and redraw the patient, but there may not be that may not be easily accomplished because the patient might have to come back in. So there's decreased satisfaction from the patient because of being, put through the runaround, and there's frustration both from the providers and the physicians because of that. Using a very conservative estimate of $25 a test, a simple, a single rejection of a batch of 48 is $1,200, and the more you do that, the more that accumulates over time. And that's just the cost of reagents, not including the cost of your time. So I think we can see that the efficiency and the cost impact of these errors can be pretty significant in a variety of different ways. And so what we really want to do is figure out where these errors are occurring. Do we have mechanisms in place to detect them? And can we improve our ability to prevent them from happening in the first place? Now, we have some structure, as you know, that's put in place by regulatory agencies like CAP that provide us with guidance on how to set up our quality assurance programs. And I'll show you some examples from CAP about what they say, and I'm sure you're all familiar with this. For those labs that don't use CAP, similar structure and clear requirements, similar structure in ISO requirements. But CAP requires that controls need to be run for both quantitative and qualitative assays. Controls are run at least daily. CAP also requires that labs set control limits, but labs have latitude. The language in the guide and the regulations say acceptability limits are defined for all control procedures, for control materials and standards, and that these controls need to be appropriate for the range of sensitivities tested, and ideally should focus on values close to the clinical decision points. But again, it says that you need to establish acceptability limits, but it doesn't give you criteria for establishing them. CAP also says that labs are required to monitor control variants and trends, but again, no guidance is provided. So we need to calculate at least monthly for quantitative assays, control statistics, and to define our analytical imprecision and monitor trends over time. But what we do with those trends, how we respond to them, what thresholds for action need to be established are not defined. So in summary, we need to run controls at regular intervals. They must have little variation in viral copies and be stable over time, must behave similarly to patients on the assay. We need to assess these values daily against some rules for acceptability, and we need to monitor values over time to detect trends and results. So what might this look like? So this is an example of an assay in stable operation. So it's a low positive control for a CMV viral load. And the middle line is the mean that was established for that control. And the hashes on either side of the mean, the hash lines on either side of the mean represent 1, 2, and 3 standard deviations away from that mean. And you can see that all the values are consistently below 3 standard deviations and mostly below 2 standard deviations from the mean. Here's an example of a similar assessment for HIV assay where there were some values that were out of range. You can see this first red box represents a violation of a 2-2S quality control rule or Westgard rule, where there were 2 consecutive control values greater than 2 standard deviations above the mean. This red dot or box represents a violation of a 1-3S rule, and that is a single value that's greater than 3 standard deviations above the mean. So the rhetorical question here is, should one, both, or neither of those runs be rejected? How we answer that question? I hope to provide you with some information that will inform you to be able to answer that question. So for the next part of this presentation, I'm going to describe some results from a study that we performed and really introduce some more sophisticated statistical analyses that we were able to perform using Unity. So we may not be familiar with the concept of total allowable error, how it's applied in a molecular setting, and I'm going to explain that to you. So this study was published in January in JCM. It was a collaboration between colleagues of mine at Bio-Rad and Mike Bachman and folks in the lab at University of Michigan. And really, at a fundamental level, what we wanted to do is answer the question, are these assays, CMV viral load, EBV viral load, and HIV viral load that were being used at the time in the lab, are they performing at a level that supports clinical decision-making? In other words, is the amount of variability that we're seeing in these assays less than what is considered a clinically significant change? So for this study, the viral load assays were being performed on Abbott M2000. CMV and HIV were Abbott assays that are IVDs, and the EBV assay is an LDT. The data collected was approximately 3 months of QC data from these assays and was entered into Unity real time. And this reflected negative, low positive, and high positive control values for each of these. Unity is a software tool from Bio-Rad that can be used to capture and analyze QC data. It has a long history of use in the core lab and is just beginning to be used for management of molecular QC data. Here we used it to calculate means, standard deviations, coefficients of variations for the data set, and also used a module in the program called Westgard Advisor that allowed us to determine the sigma level or error rate of each assay and identify Westgard rules appropriate for each assay based on the sigma value, the sigma level or sigma value for those tests. So this concept of total allowable error, TEA is really the amount of change in a quantitative value that corresponds to a clinically significant change. In other words, it's the tolerance limit of an assay. Ideally, it's based on clinical guidelines where based on clinical studies and use of these values over time and following patients in different stages of disease and in response to therapy, we've been able to establish criteria for what constitutes a clinically significant change. The TEA for an assay is comprised of both the imprecision or random errors, as well as any bias that may be present in an assay or systemic error. Exceeding the TEA, meaning having more variability than the TEA, will cause erroneous results that can impact patient care. So as an example, first example, well understood example, is that HIV has a long history in recognizing that the total allowable error or clinically significant change for that test is 0.5 logs. Where that comes from is the recognition that virologic failure of treatment is defined as something as a value greater than 200 copies per mil. And again, reminding you that individual values above that are detected require repeat testing since blips can occur. And the limit of detection in the HIV assay that we use is 40 copies per mil. So that's a 5-fold change. So your assay must be able to reliably distinguish between this cutoff of 40 and greater than 200. So mathematically, as a 5-fold change, logarithmically, that's a 0.7 log change. But guidelines that have been established by the federal government have, and through clinical trials, have established a more strict cutoff of 0.5 log relative change in a patient sample as clinically significant. So that's what we used as TEA for our study purposes. CMV, there's a little less information. Generally, what is utilized is a relative change. There's some guidelines for CMV in solid organ transplant, where the clinical guidelines have stated that the precision of the assay needs to be such that changes would be at least threefold or 0.5 logs to represent a biologically important change in viral replication. However, these same guidelines, which are cited below, acknowledge that there's a lot of variability in assays for values lower than 1,000 IUs per mil, such that a 5-fold or 0.7 log change need to be considered significant. So for purposes of this study that I'm describing, we chose to use 0.5 logs for values greater than 3 log IUs per mil as a clinically significant change. EBV, there's even less information. So as you know, EBV can be used to monitor viral loads for post-transplant lymphoproliferative disorder and other EBV-driven diseases, but there's no clear guidance on how it's utilized and how it's interpreted. There's also different information and different practice as far as utilization practice as far as specimens of choice, whether it's plasma or viral load, and that can result in differences in values that are obtained from different assays within patient and also between laboratories. The current state that's utilized is reporting the use of relative change over time, but there aren't any clinical criteria for how much of a change needs to be considered significant. So for purposes of this study, we chose to use 0.5 log to hold it to the same standard and see how our EBV assay performed in that context. So next, I'm going to introduce the concept of sigma values for molecular, because we now understand the clinical limits under which these assays are performed, and we can now apply the concept of error rates into this context. So you may be familiar with sigma values as they're applied to lean principles in error detection and processes. So sigma metrics are used to quantify how often variations occur in a process that can lead to a defect. And those errors are measured as defects per million in a process. So if you're manufacturing a part to a certain specification, the sigma value tells you how often is it so far out of spec that you can't use it or that it malfunctions. In this context, the standard is that 6 sigma is world-class quality, and that's equivalent to less than 3.4 defects per million products, occurrences, or procedures. In a lab, though, you may not know that you put out an erroneous result in a patient sample, from a patient sample. And so we need to infer this from our control values and monitor error rates and controls. For a laboratory test, sigma accounts for imprecision and bias, and you calculate the sigma value by using the total allowable error expressed as a percent minus any bias in your assay divided by the coefficient of variation expressed as a percent. And so the TEA and CV and bias are all relative to an assigned control value. Bias is a difficult thing to measure. One way you can measure it is by comparing your results to a peer group. If you know all of them are using the same method, the same platform. You could also attempt to quantify bias through comparison of your results to proficiency testing results. So it's really important to understand bias in assays, especially if patients are being tested across multiple labs. This can cause errors to occur in one direction or another. You could have a positive bias and over quantify in one assay, or a negative bias and under quantify. For our analysis, though, we focused on within lab performance with a single pathway for testing, instrument pathway for testing, and assumed that there was no bias for purposes of our calculation. So if you redo this equation, removing bias, it's really TEA as a percent relative to a mean divided by your coefficient of variation as a percent. So to share some study data, as a reminder, we collected data for low positive and high positive controls. The data shown here is for low positive controls for each of the assays. And you can see that calculated mean for those 3 months of analysis are shown here for each of the values. Standard deviations, coefficient of variation, all really good. So if we calculate our TEA for each of these assays, I told you that we were using 0.5 logs per copies per mil, or IUs per mil, as the TEA. As a percent, it's that 0.5 divided by the mean times 100. So TEA as a percent is shown here. And then to calculate sigma, you divide TEA by your CV. And so this is where we get the sigma values. So we got for CMV a sigma value of 11, for EBV a sigma value of a little over 5, and for HIV a sigma value over 0.7. So remembering that I said 6 is world-class performance, 2 of the assays were well above that, and 1 of the assays was slightly below that. But if you look at the confidence intervals for these, you can see that the amount of change that would need to be detected with 95 and 99% confidence is still well under 0.5 logs. So even for the assay with the lowest sigma value, we're still detecting change of 0.25 with 99% confidence. So still really good performance. But if we look at this kind of, I'll say in air quotes, the worst performing test, EBV, and this example for the low positive control, that sigma value of 5.06 is 200 is equivalent to 230 false results per million tests performed, or 1 false result in every 4,300 or so individual patient tests. And based on the testing volume at Michigan Medicine during the time of the study, that would be 1 false result every 1.25 years in that lab. And that's if no QC rules were in place. So it's just taking the values that you got and accepting them and reporting the results. We would have had 1 error every 1.25 years. And reminding you that this is performance of this is assay performance for an LDT. EBV was the LDT in this example, or in this study. So for the other 2, the IVD assays, that sigma values, again, were extremely high. So that sigma value of 7.66 for HIV is 1 error or 1 false result in every 2.78 billion tests. And for CMV, that's 1 in every 10 to the 14th individual patient tests. So these assays perform very, very well and were highly unlikely to result in any clinically impactful errors. So this graph that I showed you before was actually the HIV data from the data set, and reminding you that we have the mean for this data set and 1, 2, and 3 standard deviations here. What I didn't point out is the numbers on the right actually reflect the plus or minus 0.5 logs, or the TEA. And so you can see that these errors that were flagged based on the Westgard rule, even though there was variability and it generated a flag, the variability was still well within the TEA of the assay. So the previous data adds meaning and context to how we can establish QC rules or Westgard rules. And I'm going to show you how we used the program to calculate those rules and select the rules to apply based on the amount of variability we were seeing in the test. So to do this, what do you need to know? You need to have your control values over time. You need to know your TEA and present it as a percent of your control mean. And in our case, we used Westgard Advisor inputting this data into Unity to calculate that for us. So I'm going to show you some screenshots on how this works. So here we've selected HIV as the assay that we want to calculate our QC rules for. When we select on that control, it has the user-defined laboratory rules that were in place in the lab at the time. So the lab was using 1,3S and 2,2S as warnings or flags, maybe yellow lights, and anything that was 1,4S was considered a rejection. It's also showing you the data for the time relevant to that lot of controls and how many data points are in there and what the summary statistics for that are. Once you have that information, you can select Configure TEA and tell the program that you're putting in your user-defined TEA as a percent, and it's calculated again based on that 0.5 divided by the mean times 100. And so the program will go through and make calculations and tell you based on the TEA what your sigma value is, what your existing rejection rule is, and it says based on the amount of variability that you're seeing, we suggest using a 1,5S rule, so a less stringent rule than the laboratory was using. So if you look a little bit further into this data, and I'll ask you just to focus on the top up here, the 1,4S rule was what the lab was using. The program suggested 1,5S, and it's likely because there was a demonstrable difference in the false rejection rate between the two at lower decimal points that we can't see. But the PED, or probability of error detection, actually showed for the laboratory's purposes a much more clinically relevant difference. And so the suggested rule of 1,5S had a probability of error detection of just about 94%, but the existing laboratory rule had probability of error detection as almost 100%. And so based on that, that higher PED for the 1,4S rule and essentially the same ability to detect false rejections, the lab chose in this case to stick with the 1,4S rule. So using EBV as an example, I wanted to show you the impact of utilizing different rules. And so our goal is error detection of 90%. So if you would apply a very strict rule of 1,2S, where anytime a 1,2S rule is violated, running three levels of control, negative, low positive, and high positive, using the existing data, the 1,2S rule would calculate or detect errors 100% of the time. But with this very strict rule, it would have a false rejection rate of almost 13 or over 13%. So that's a false rejection in more than 1 out of every 10 QC results. So you can see how that's represented in the graph. The PED is 100, and the probability of false rejection, PFR, is 13.1. So I think we would all agree that that rule is a little too strict and we're rejecting too many runs. So if we compare a couple of other rules, a 1.3S rule versus a 3.5S rule. So with that same data set, if we applied a 3.5S rule, we detect errors only 86% of the time, but our false rejection rate would be 0.1% or less than one false rejection per year. So I think the happy medium here is a 1.3S rule where we're detecting errors 96, almost 97% of the time, and still having a very low, less than 1% error, false rejection rate. So while that's a higher chance of detecting, it's a higher chance of detecting errors, but only 2 false rejections per year based on laboratory volume. So that was seen and recognized as the ideal rule to use based on the amount of variability for the EBV assay. So the data that I showed you up to this point was limited to the 3-month study period. I showed you examples of low positive control data. We calculated the same for the same data set. We looked at the high positive control values and saw exactly the same type of information. Very, very high sigma values for both CMV and HIV, lower sigma value for EBV, but still very high. You know, it's important to recognize that that's just, you know, one snapshot in time with a single lot of reagents. So Dr. Bachman went back and applied the same kinds of calculations to different lots of controls for each of these assays tested over time. And so this is data for HIV with different lots spanning 3 to 4 years, 7 lots of reagents through over 400 control values and 5 years. And the mean sigma for the low positive was over 7, and for the high positive was over 8. So still consistently well performing. Based on those rules, Westgard suggested 1.5S, but the lab decided to be a little more conservative and used 1.4S rule. For CMV, there's been a little bit of variability in this test. The 11.7 sigma value was pretty much an outlier from the lot that we were using, but it's settling into some stability in its variability with greater than 7. So the mean sigma for all these lots was well over 7. For the high positive control, it was over 9. So just reinforcing that it's important to test multiple lots to see whether or not the patterns you're detecting are consistent. So when the lab did this assessment, it really showed that the assays were performing very well. In the context of for the IVD assays, manufacturers' QC ranges that are really, really wide. And so those, you know, I think objectively, those very wide ranges are not designed to monitor assay performance over time. They're really designed to detect any catastrophic systematic failures in your testing system. And so the analyses that I've described can tighten that window down and hopefully allow you to see that any trends or abnormalities that are potentially occurring. What they found in the lab when they started implementing these rules based on this analysis is that what they were finding were really operational issues, such as the testing personnel failing to record new lots of reagents or controls. And so there were unexpected failures because the values that were being detected were different than what was expected for the lot being used. And there's also failure to record instrument validation or repair, which resulted in some variations in trends. And it also showed that there was inter-user variability in how controls were handled in the lab. So for well-performing assays, such as described, what they were primarily detecting were documentation failures and not necessarily clinically significant changes in performance analytically of the test. But those documentation failures could result in false rejections or false acceptances. So I don't want to minimize the significance of those issues, and it just underlies the various sources of errors that can occur and the importance of having a very robust QA program to identify them. At the end of the day, though, if your sigma values are really good enough, it's likely okay that the manufacturer's QC ranges are high, but you can't just assume that without looking at the data and calculating those sigma values. So just to wrap up, a few things that are important to remember is that I feel like you really need to work with your clinicians to understand what their expectations on performance are, and knowing the total allowable error and being very clear both what you can measure and what clinicians expect is really, really important. And unfortunately, it's not known for all of our assays. I think it's also really important to look at the variation, and determining sigma values is a way to provide you with information on whether your assays are meeting these clinical expectations. And quality control or Westgard rules can be set loose or tight based on how variable your assay is relative to the TEA. And Unity, I hope I've shown you, Unity Westgard Advisor can help you with that process as a tool. But regardless of whether you use Unity, I feel like you have to have a robust mechanism to calculate these statistics and assess the performance of your assay. And ultimately, it's a balance of the cost of false results with the cost of false rejection. And so finding that sweet spot in error detection and false rejection is really important. I think what I've shared with you today is kind of a first step in establishing what I feel like are robust processes for QC data management. And it starts to build the foundation for what I feel like are more in-depth question or higher level types of activities that could be considered. And so one of the things to think about in this context is the value of adding an external or independent control, something that has material or something that has a long shelf life with a known value that's well characterized, to help move us in a direction towards portability of assays. So if you're running these independent controls over time across your assays and multiple labs are using the same material, it makes a comparison of assay performance between labs conceptually much easier. I think one of the things we also need to be aware of and spend time on is not only assessing, but accounting for the biases that may be present in our tests. So new calibrations and new lots may shift the values of our assays over time, and we may need to alter the rules that we use for acceptability or rejection based on changes in bias over time. And again, based on the first statement I made on the previous slide and the communication with our clinicians, we need to be aware of changes that occur in clinical practice. And it's important to incorporate clinical guidelines for any of our viral load assays and how they're being used as they emerge over time or are refined over time, so that our interpretations and our assessment of what's acceptable is accommodated by that. So I want to thank you all for your attention. I really want to thank the folks from the Clinical Micro Lab at Michigan, Mike Bachman, who's the director of the lab, Arthi Raman, who's the QC supervisor, Courtney Branchow, senior tech, Jacob Abel, who is a molecular fellow at the time, my colleagues at Bio-Rad, who were instrumental in helping with the study design and calculating the data, Nico Vandepol, John Pacheco, and David Gauthier. I'm really happy to take any questions at this time, and thank you very much for your attention. And I apologize for the pop-up windows. I thought I had turned everything off, but apparently I'm a boomer and I can't figure out how to use my computer. So thanks.
Thank you so much, Yuen. This was fantastic. The way you simplified those complex QC rules, and this has been fantastic talk. We have a few minutes for some questions, so I encourage everybody to type the questions in the questions and answer or chat. But there is a question about thinking about how to implement Unity in the clinical lab, and what do you think can be really the model here, like in the core lab where this has been used. They usually run the QC first, and if this passes, then they run the specimens. How do you think this model fits in the molecular biology lab?
Yeah, I think one of the things that we've been trying at Bio-Rad have been trying to spend time on, and me, from a scientific standpoint, is understanding how to apply these concepts in a molecular lab compared to chemistry. And the reality is that the approach that we take in molecular is different. And we're running controls with each batch of patient samples, whereas the core lab is running controls and then subsequently running samples continuously over time. And so repeating controls at different points during the day is really important to determine whether or not any variation is happening. So I think the reality is that we don't have as many opportunities to see variation because we have batches of data periodically. And so I feel like, to answer the question, I feel like we have to look at our controls for long periods of time and get lots of data to understand, and what lots is, I don't know. Statistically, more is better, but I think as we start doing this, using whatever tool we have at our disposal, and as we start doing this, I think it's really, really important to do these calculations so that we know how our assays are performing. And so we have to start somewhere, and then by doing that, we're accumulating a body of data that informs us as to how our assay is performing. I think it's very reasonable conceptually to incorporate an independent control that's separate from your manufacturer's control, that has a longer shelf life, longer out date. So you're gonna get new lots every 6 months, but if you have an independent control that's got a 3 or 4 year out date that you're running and able to compare different lots controls to over time, that can help you with this assessment as, is this new lot acceptable or not? I think that adds value as well. So that the two parts to it are, know what your assay is doing over time. And for us, time is months. For, the core lab, time is hours.
Right.
So I think that's the biggest difference.
Right. So do you think then this should be, will this be different between lab developed test versus IVD, or what do you think?
I think, in my mind going forward, I didn't know. I think I didn't trust until looking at, doing the study. I didn't trust that the manufacturer's wide ranges of acceptability were appropriate. I think it's still important to recognize that the controls from different manufacturers are formulated in different ways and don't necessarily reflect patient samples. and are not, you need to really scrutinize their characteristics and attributes to ensure that those controls are really controlling for all aspects of your test. So, I think that's the first thing to look at, whether you have an IVD or an LDT. And if your controls are, robust and are controlling for all aspects of your test, I think, our data showed that the IVDs perform better than this particular LDT. But I think I would expect, and I would hope that others in the audience would be inspired to do the same types of analysis, and we could generate more data to see if that's true. And, not only comparing, other Abbott users to what we generated, but Abbott to Roche to Hologic and other manufacturers. I think I don't really know the answer to that question. I think it's theoretical that the IVDs are going to perform better, but I think it's too soon yet because we don't have enough labs doing this type of analysis. So, I'm kind of being a Pied Piper to say, I think we need to do more of this.
Well, unfortunately, we are at the top of the hour, but this was a fantastic session. Thank you so much, Wayne. And at the conclusion of this webinar, I really would like to really thank you for sharing your time and expertise in this area, which is, as I mentioned, it's one of the most important, most complicated aspects in the clinical or molecular virology lab specifically. As a reminder to everybody though, we have a couple of webinars that are coming soon. We have a neonatal CMV screening and the diagnosis. This is in 2 weeks. And 2 weeks after that, we have a discussion about the picornaviridae, surges and antiretrovirus D68, parechovirus, and a discussion on poliomyelitis. And please visit our website for more information and also to register. And at the end, I would like to just remind you to please get your influenza and COVID booster shots to stay safe, especially before Thanksgiving. And happy Thanksgiving, everybody.
Thank you.
Yeah.